Hello, everyone. Welcome back. Thank you for joining us. We're glad you're here. I'm Steve,
And I'm Scott.
and together we're Backyard Musings.
Yep, we're glad you're back with us, and we're talking about a another cancer topic. We talked about some of those last week, and this is another notice. So here we go.
Yeah, and this pancreatic cancer causes, so much destruction, and it's hard to trace. Okay, here we
And
go.
treat, yeah.
Researchers found that nerves and cancer-supporting cells team up early to drive pancreatic cancer. Breaking that alliance could weaken the disease before it fully forms. Pancreatic cancer is one of the most hardest cancers to find early and one of the most difficult to treat. Standard therapies often fail, which has pushed scientists to search for new ways to interrupt how the disease takes hold. While researchers have long known that the nervous system can help cancer spread later on, its role at the very beginning of pancreatic cancer has been much less clear. Would you like to know more?
Okay, starting off with a quote here. One phenomena that is known is called perineural invasion, says Jeremy Nigri, a postdoc in Professor David Tuvison's lab at Cold Spring Harbor Laboratory. I don't know where that is, but it sounds fun. Quote again, this means cancer cells will migrate within the nerve and use the nerve as a way to metastasize. Unquote.
Yep, maybe kind of like a little highway, to spread its little things. Crazy.
Yeah.
New research from Negri and his colleagues shows that nerves are involved far earlier than previously thought. Their work reveals that the nervous system actively contributes to pancreatic cancer development even before tumors appear. Using advanced 3D imaging, the team found that tumor supporting fibroblasts, known as MyCAFs, MyCAFs, plural, MyCAFs, release signals that draw in nearby nerve fibers.
Once these nerve fibers arrive, they interact closely with MyCAFs inside pancreatic lesions. Together, they help create conditions that allow cancerous changes to progress. The study was published in Cancer Discovery, a journal of the American Association for Cancer Research.
To uncover this process, the researchers used a method called whole mount immunofluorescence, which allowed them to capture three-dimensional images of pancreatic lesions and the cells surrounding them. Traditional 2D images show nerve fibers as small disconnected dots. In contrast, the 3D images expose a thick interconnected web of nerves winding through and around the MyCAFs and lesions.
Quote again, when we first saw this picture, I was shocked, Nigri said. I couldn't even imagine the lesions like this. I'd only ever seen it in 2D. Further experiments using mice and human cells revealed a powerful feedback loop between MyCAFs and the nervous system. The researchers found that MyCAFs release chemical signals that attract nerve fibers from the symptomatic nervous system. Is that right? Symptomatic?
Sympathetic.
Sympathetic, sorry. Which controls the body's fight or flight response.
Huh. These incoming nerve fibers release norepinephrine. Yeah, I said that right. Norepinephrine, a neurotransmitter that binds to fibroblasts and triggers a surge of calcium inside the cells. This calcium spike further activates MyCAFs, encouraging precancerous growth. At the same time, it draws in even more nerve fibers, strengthening a cycle that continues to fuel disease progression.
The team also tested what happens when this nerve activity is interrupted. Quote again, in one experiment, we used a neurotoxin to disable the sympathetic nervous system, Nigri says. We show reduced fibroblast activation and a nearly 50% reduction in the tumor growth.
Wow, that's good.
Yeah.
That's encouraging. Because this nerve fibroblast interaction occurs so early, targeting it could open the door to new treatment approaches. The findings suggest that existing drugs, including doxycycline, and we're not a sponsor of that. They're not a sponsor. Might be useful when paired with established therapies such as chemotherapy or immunotherapy.
Finishing off with a quote here. The next step will be to study this more in detail and to try to find way to block the crosstalk between fibroblasts and nerves, Nigri said. Quote again, going on with the quote, with support from groups like the Lustgarten Foundation and Pancreatic Cancer Action Network, we hope to one day help improve patients' outcomes. That's a good thing if they can do that.
I wonder, I mean, when do they start this, because it's hard to detect pancreatic cancer. I could see it starting at different stages,
Yeah, it almost seems like when people find out that they have this, it's too late.
Too late.
It's too late for any kind of treatment. So I don't know. But I know that thing that we talked about last week, that sounded very promising. I can't remember exactly what the issue was, but it involved pancreatic cancer. And that sounded promising too. So these guys, it's good to know that these guys are working on it. But again, I think I mentioned it last week. It's been years and millions and millions and millions of dollars invested in these researches, and you'd think that they'd be able to come up with something. But 50% reduction in tumor growth—that's pretty good.
We talked that the different healthcare things need to communicate, into like a database,
Yeah.
We talked about that.
Yes, we did. And that we would hope that's happening, but let's see.
All right, folks. Thanks for joining us. We'll catch you in the morning.
Take care. We'll see you tomorrow. Have a good rest of your day.